Transcription is the first stage of the expression of genes into proteins. DNA is transcribed by the enzyme RNA polymerase. This enzyme initiates transcription, joins the RNA nucleotides together, and terminates transcription. To initiate transcription in bacteria, a variety of proteins called sigma factors bind to the RNA polymerases. This complex can then bind into a specific sequence of usually about 40 deoxyribonucleotide bases, called the promoter, located along the DNA prior to the coding region of the gene. The promoter determines what region of the DNA at which strands of DNA will be transcribed into RNA. Transcription may be broken into 5 stages: pre-initiation, initiation, promoter clearance, elongation, and termination. The first step of transcription is called pre-initiation. RNA polymerase and cofactors bind to DNA and unwind it, creating an initiation bubble. There is a space that grants RNA polymerase access to a single strand of the DNA molecule. The initiation of transcription in bacteria begins with the binding of RNA polymerase to the promoter in DNA. Transcription initiation is more complex in eukaryotes, where a group of proteins called transcription factors mediate the binding of RNA polymerase and the initiation of transcription. RNA polymerase must clear the promoter once the first bond has been synthesized. Approximately 23 nucleotides must be synthesized before RNA polymerase loses its tendency to slip away and prematurely release the RNA transcript. With elongation, one strand of DNA serves as a template for RNA synthesis, but multiple rounds of transcription may occur so that many copies of a gene may be produced. Termination is the final step of transcription. Termination results in the release of newly synthesized mRNA from the elongation complex.
Translation begins when a ribosome connects onto a start codon of an mRNA molecule in the cytoplasm. Next, tRNA molecules attached to amino acids dock at the corresponding codon sequence on the RNA molecule. This process repeats over and over, with multiple tRNA docking and connecting amino acids into a growing train that goes out on top of the ribosome. When the ribosome comes across a stop codon, it falls off the mRNA molecule and releases the protein for use in the cell.